Good morning and welcome to our fourth seminar for the 10th series of International Plant Breeding Seminar Series. It is a pleasure to have with us Dr. Hewan from Ethiopia. Hewan holds a PhD in bioresources engineering, but has a robust academic foundation in molecular biology, genetics, and biochemistry. She is the Associate Dean of Research and Technology Transfer at Hawassa University in Ethiopia, and she focuses on improving crops to enhance food security and nutrition. Most of her work nowadays focuses on a big project to improve barley resilience to acid soil stress, and I think that's going to be the focus of her presentation. Hewan, it's a pleasure to have you with us. We look forward to your presentation. I'm going to stop sharing. I understand you have a brief introductory slideshow, and then you go to your presentation. Thank you so much. I'll give you the floor.
Thank you very much, Carlos. Now I'll share my slide just a little bit to show you how barley looks like in Ethiopia.
Sure.
So my title is Translating Molecular Research into Agricultural Impact. This research I have started after joining Hawassa University because I have realized that there is acid problem in the region where I am working. And I collected different type of barley from different parts of southern region of Ethiopia. Because barley is one of the deer crop in Ethiopia where we use it to celebrate life. Every woman after giving birth, she invited her relatives, her neighbors, and prepare a porridge like big feast, a big feast with a porridge, and they celebrate the birth of a newborn. Barley is versatile in Ethiopia, where it is domesticated purposefully for food, feed, and also drinks. And each farmer, they have their own type of barley. In each village, they collect the seeds from the farmland and they keep it for the next generation. So we don't have a seed procurement system in our country still. I think in the future it might happen, but the gene bank for the farmer is the farmer himself. So they collect different type of barley and they use it to make different type of dishes. So all this diversity, the way they selected, it created a unique barley from the rest of the world. So why it become unique was my question. And in relation to the current problems that we are facing in our region with acid. So I am connecting that to the problem of climate change and the losing of productivity for barley, and specifically in the highland region of in the southern part of Ethiopia, where barley can be the only cereal which they use for different type of their traditional foods and feeds. So the current scenario of acid soil enforces us to breed acid tolerant barley. So for that, we have to search if there is acid tolerant barley in Ethiopia. And the good thing that we have collaborated with Okayama University from the Institute of Plant Resource Center, Stress and Resource Center, Okayama University, which is located in Kurashiki, with Professor Sato, now is succeeded by Professor Hisano. We have seen how devastating is the acid problem in Ethiopia, and we devised to breed acid tolerant barley for Ethiopia. So as you can see, there are different type of barley for the farmers, but if these barleys are gone, the custom, the culture of these people might be affected. So that's why we stop there in breeding acid tolerant barley for that. So I will share with you my other screen. Sorry. Just a moment, please. Thank you very much. So as you see, my title is Translating Molecular Research into Agricultural Impact, which taught me a lot of lesson and also helped me to work with the farmers and also the masters and also PhD students in our university. So why I choose that is because climate change, it affects the energy in the world, which according to the IEA, we need 50% of increase in the energy and the availability of water and also food from the FAO information. So we have to increase food by 50%. There is a demand. So this has to link with the different agroecologies, and the highland ecology is one of the agroecology where barley is the only cereals, and it is currently affected by different climatic condition. One is the soil pH. The soil acidity is drastically increasing. As you can see, this map that is courtesy from Leon Kochan, as you can see from North America to South America, and about this, Central Africa and also Eastern Africa, for Russia and Asia and also Australia, all of these regions are really affected by acidic soil. So this acid, it has affect the productivity of different type of plants, and specifically with the crops plants. And crop plants, they have different mechanism in order to tolerate or be susceptible to the acidic soil condition. So when we look at in Ethiopia, the map, as you can see here, shows that in the southwestern part region, the majority of the land is acidic, from strong acidic, which is the red one, and also moderately acidic, which is colored here by yellow. So this acidic soil, what is this effect then? Because the acidity of the soil, it has something to do with the genesis of the soil. The major reason for soil acidity in our region is because of the plant material. As a plant material, it is composed of ignimbrite or basaltic procyclic deposits, and this genesis result in reddish type of soil. So when you look at the soil, it is red. So it shows you there is high accumulation of iron and high aluminum oxide. So the aluminum oxide, especially the aluminum ion, is dominant actually, which affects the growth of cereal in that particular region. And this region in the southern part of Ethiopia, it has heavy rainfall. So the heavy rainfall results into leaching the land and taking away the nutrients by the erosion, and also the farmers' practice of use of acidic forming fertilizer, like for example, nitrate, which react with the acid, the rain, or like in the water, it forms nitrate, and it removes the base elements, increasing the number of basic elements being removed from that place. The other characteristic is the use of high yielding barley lines. So a repeated cultivation of the barley lines in that particular area, it creates the taking away of all the organic molecules or the inorganic and organic molecules, which is really important for the soil fertility. And also sometimes, like, you know, the straw is decomposed in that particular region, which increases hydrogen toxicity, which releases hydrogen ion. Actually, it affects it reacts with carbon dioxide and it forms organic acid, which aggravates the replacement of the bases from that area. So all these different scenarios affected the soil in that particular region in the southern part of Ethiopia, where acidity became terrible for the cultivation of barley. So how acid affected the growth of barley is because it is related with the increasing or decreasing of soil pH. It makes the increasing or decreasing of available elements. So with the simple example here, you can see from this figure, when the soil pH, when the acidity increases, some of these elements like iron, manganese, copper, zinc, and aluminum become abundant. While when the soil pH increases, it become lower in the soil concentration. So they become like, you know, ion in the acidic soil environment. That creates a reaction with the important nutrients like nitrogen, sulfur, and potassium, and also molybdenum and phosphate, where their availability is determined again with the pH. For example, molybdenum become lower in the soil, in acidic soil. That is also important for nitrogen fixation or for the other. And phosphate, it is optimum availability in the soil is at a neutral pH, where around 6.5 pH, the phosphate become available. So that means available for the plant to absorb, to be absorbed by the root system. But above that, what will happen, even if it is available, the plants, they will not be absorbing it. And so it affects the nutrient, which in turn affects the yield or the productivity of that particular plant. So the presence of soil acidity, it disturbs the availability of essential minerals and also elements for the growth of any type of plants in that particular region. So when we come to the response of living organisms, if humans are stressed with acid, yeah, they just move from one place to the other. But plants are permanent in that place. So they can't move and they have to adapt to that particular environment. So the adaptation of the plant in that particular environment, specifically in acidic soil, is different. For example, in our research also, insect is one of the orphan crop where 20 million people in the southern part of Ethiopia, they are using it, but we haven't heard the problem of acidity. Why we didn't hear or such kind of information about problem of acid on insect? Because insect is a home garden, which is nourished by the cow dung and cow feces, actually, which neutralizes the soil acidity in that particular region. And kale is also another type of home garden crop, which we don't have a lot of information about the acidic effect on kale productivity. But barley is highly susceptible. So as you can see, all of these living organisms, including insect, kale, and barley, they have different genome, from 46 chromosome human being to 18 chromosome in insect, to 18 chromosome in kale, and also 14 chromosome in barley. So what will be the effect then? So they must have some kind of mechanism in order to tolerate this particular acidic soil and their growth and productivity. So when we look at among the cereals, barley is highly susceptible or sensitive for acidity. In the case of maize, most of the time it grows in the warm area in Ethiopia, So we didn't hear a lot of information about the effect of acid on maize. But in the case of finger millet, one of the orphan crop in Ethiopia, which is also originated in Ethiopia, it is highly tolerant for acidic soil. So you might ask me then, and then why the farmers, they are not using the finger millet rather than sticking into the barley in the highlands? That is because finger millet, most of the time, it grows in the lowland area, and the selection hasn't been done so far. So barley dominates as a cereal crop in the highland area. When I say highland area in Ethiopia, it's about above 2,000 meter above sea level, and that is acidic soil in the majority of the southern part of Ethiopia. So to unravel the mechanism, to understand what is really happening with barley, we have to go back. It is origin of domestication. Actually, this paper, which I am showing you right now, this figure is published with my collaborator as a co-author from Okayawa University. And they showed from their research that barley is started domestication 12,000 years ago in the Fertile Crescent in the northern Levant and southern Levant region. It originated from spontaneum. And this selection of certain traits started the domestication of barley. One of the traits which is interesting is the brittleness of domesticated the wild barley compared to the domesticated barley. What happened with the wild barley? Whenever it forms a small seed, it by wind or by other forces, it scatters. It is seed, and that is how the majority of wild type species of plants are dominating the wild or the forest area or the wild environment. While in the case of domesticated barley, it has intact seed, which stays longer until it will be harvested by the human being to be used. So barley, it started domesticate the hunter-gatherer 12,000 years ago started domesticating barley by selecting the brittle gene, which they have identified at its different mechanism based on the northern Levant and southern Levant region. So I took that inspiration or that lesson from this paper, and I asked myself then, why not? How these Ethiopians have domesticated barley 9,000 years ago? Because this barley is the most important economically viable plant in the southern part in Ethiopia, about 40 billion US dollars that we can get it out of that. And it has easy agronomic managing practice, and there is highest biodiversity. In the biodiversity, we have about 16,000 different types of barley, which is accumulated in the biodiversity. And it has a better productivity in the world, and it is a resilience and actually involved in the majority of the alcohol industry. Specifically, you can mention about beer. So this diversity enabled for the selection with of different barley traits. So to mention that, the barley domestication, which I have mentioned, the BTR gene, which has been published from Okayawa University, it shows that the mutation at chromosome number 3 with of this BTR1 and BTR2 gene resulting to the mutant line BTR1 or BTR2, and in the form of heterozygous formation, the brittleness of this barley loses and it becomes intact. That is number one of the barley domestication. The other one is all wild barley are true raw barley, but in the case of domesticated barley, we find this 6-row barley. So what happened to that? Because of the selection of this BRC1 gene, which which is HD zip transcription factor, which is found on chromosome number 2. So the row number gene is changed as a functional allele changed, and it increases higher yield. The 6-row barley has higher yield, while 2-row barley is lower yield, but it has a good malting quality. So barley domestication number 3 gene is the nudity. And after 9,000 years, barley become naked. Why? Because the nude gene, ERF transcription factor, which is located in chromosome number 7, created a hairless and hulled or naked grain barley. And this naked barley is easy to process for food, but the hulled barley, it is better for protection if you are keeping it for longer period of time. So such kind of domestication is really going on. Number 4, the vernalization gene, which is divide the spring versus the winter grows barley. So for example, for us, most of the time we plant barley in 2 seasons in Ethiopia. But there is a winter barley in some regions, and specifically those who are living in the northern hemisphere that can understand that. That is because of the vernalization gene is playing a role in flowering time. And the other gene is the photoperiod gene. This photoperiod gene, the mutation in the photoperiod resulted into photoperiod sensitivity in early flowering plants, which has a wider adaptation, but the sensitive alleles, they are only flowers in the long day, which have a limited adaptation. So all that selection resulted into to develop or to have different type of barley. So my question was that, okay, then what was the selection factor? What did the Ethiopian, they have domesticated? Because when we look at the culture, we use barley for making different type of food for human being. And we also make alcoholic drinks. And also the straw is used for the house covering, for specifically for making tukul. And also the straw is used for feed, animal feed. So that means there is nothing that we throw away from barley. Everything is usable in the Ethiopian community. I think the Ethiopian, they have selected a specific type of barley in the domestication syndrome. So why I become bold to say like this is because when we gather data from the southern part of Ethiopia, and with different ethnic group, they informed us why they have a specific type of barley in their hand. Most of the farmers that we have asked them, they have greater than 3 barley land races on their hand. And even some of the wealthy people, they can have more than 6 barley in their hand. And they tell you what each different type of barley, what type of characteristics they have, and they describe for you. For example, I will take you in one of the ethnic group in the southern part of Ethiopia, that is the Gurage ethnic group. So when we ask these people, they tell us each of the barley, it has a name. Some of them, they call, one of the barley is Jumatukur. It is 6 row, and it is black. And they say that it is short spike black seed. And they selected that it is early maturing, and it has high yield, and it is drought and disease tolerant. But they are not happy because of the blackness of this barley, because most of the farmers, they prefer white barley. So it has low market demand, but the farmers, they use it for the household consumption. The other one is Audo. They are really happy when they talk about Audo. And this Audo, it has two row spike, and the seed is white, and it has a white kernel. It's very good for market, and they call it is the best food quality. And the yield is, the problem is the yield is intermediate. And the other one is what the Agriculture Research Center, they released for them, one of the variety, which they call it shaggy. And it is 6 row, it is high yield, they are happy with that, and it is white, they are happy with that. But they say that, oh no, we don't want it for food in our consumption. Usually, they sell it for the city people because they think that it has less food quality. And the other one is Chifiaudo. And they say that it is best food quality. It's quite good, but no, the yield is not that much interesting. So you can see that a number of these different ethnic groups, they have different name for their barley, and they keep those barley with their hand and in their own house. So even if we have the biodiversity, we have the research institute, but still the farmer, they keep significant barley land races in their own house. And we collect that with their generosity. We collect all this barley, and we have identified that, yeah, farmers are preserving the land races, and their preference is a specific color, row type, drought tolerance, or disease tolerance, and the taste of the barley, that is really important. So that means the Ethiopians are selecting barley based on its taste. That implies they might be selecting based on the starch content or the protein content or the different mineral content when they are testing it. So I was not able to identify it, but after you give them certain type of barley and they tell you that this is Audo, this is Shaggy, or this is something, it's really amazing when they are describing how they identify the barley. So for that, we collected about 170 barley accessions from the southern part of Ethiopia, and we wanted to check by using molecular marker if there is significant difference, or if the farmers, what they are describing is also can be confirmed by this genome-wide SNP marker. And after cleaning the SNP, after we get it from the dart sequencing, we screen, and we have got about 3,553 SNPs, and which is covering all the 7 barley chromosome, and we use that for diversity analysis. And we have seen that based on the diversity, all these 170 land races are actually unique genotype. And the overall diversity is very high, and they have the population structure varies from one region to the other, or from one ethnic group to the other. For example, what we have really identified that in Gurage region, there is this index of association, which is the highest compared to the other part of the region. For example, in Sidama region, right now where I am living, the index of association is higher, it is about 104, but in Samin Omo, it is low, about 22.8. So there is the seed, the diversity, the inbreeding, everything really different based on the different ethnic groups. So the population of Sidama, they have high diversity, moderate population structure, but in the case of Gurage, there is high structure, and that might be because of inbreeding, because high land above 3,500 meter above sea level, it is very cold in that area. And I heard that the farmers in this part of very cold region, they don't lose one variety which fits into their environment. So for generation, they transfer it from one generation to the other, and they keep the seeds like that. So that's why they have different type of inbreeding. So as you can see from this picture or table, the genetic diversity from each ethnic group is varied. Some are diverse but not structured. Some are highly structured. So all this information that we have gathered. The other one is the regional diversity based on richness, Shannon, and Simpson index. It shows that in the case of Sidama, where I belong right now, there is rich, balanced, and low dominance of barley or diversity. But in the case of Gurage, it is higher, the richness, but there is a slight evenness because of the inbreeding is really happening. So all this information confirmed for us there is selection going on by the farmer. And from that analysis, we come to understand that there is about 3% only difference among the different ethnic groups, but there is huge diversity among the individuals when we look at it. So this tells us we confirm that each ethnic group, they select different type of barley and they keep it. And as a result, there is huge or significant genetic variation between the individuals that we use this information in order to select barley for our future purpose. So from the phylogenetic tree, you can see that even if it forms tree structure, but there is a repeated selection by the farmer, which result into introgression or admixture. As a result, there is unique or rare genotypes has been identified. So what we have done from our selection is that, okay, let us see if the selected land races, specifically from the different ethnic group, if they are really different based on the world collection of barley. And we use the barley collection from Asia, the one we collected from Gumer, the green one here, and from other parts, the northern part of Ethiopia, and from the Western world, specifically from Europe and United States of America. So all those barleys, it shows that clear structure. So we have seen that the collected barley land races show clustering, or they form a group with the spontaneum and with the Asian group, forming two structure from Ethiopia. So compared to the Asian land races, starting from Iran or up to Japan and also some regions around there, it shows that the Ethiopian barley has a population structure. At least we have got two different clusters. So this is a very good information, the presence of high level of genetic diversity within and between the population of barley in Ethiopia. So this gives us confidence, and we wanted to select acid tolerant barley from Ethiopia because barley has its own adaptive mechanism in acidic soil. And this research has been well investigated by the Institute of Plant Resource and Stress in Okayama University, that is in Kurashiki. The metal detoxification is one of the methods because barley releases organic acid through the root tip, and also metal sequestration is another method. And also they store these toxic elements with their vacuole. That is one method, metal detoxification. The other one is enhanced nutrient acquisition because of root adaptation. So we wanted to check if there is diversity based on the root adaptation, and a upregulator transporter for essential elements like phosphate and calcium and magnesium. This resulted into different mechanisms. We saw that, we hypothesized that this will affect the selection, or the different, we can use this different barley for understanding that mechanism. And oxidative stress management is also another mechanism which results into activation of antioxidant pathway to combat reactive oxygen species. So we start first from the well-known information from Japan, and they have identified that the majority of the Northeast Asian land races of barley, either they have a 1 kilobase per insertion at the HVAST, which is confirmed that the presence of 1 kilobase per insertion at the transcription start site, where at the multiple transcription start site here, it serves as a mechanism for this HVAST gene to be expressed. So those with 1 kilobase per insertion, they have confirmed that they are aluminum tolerant cultivar. That means acid-induced aluminum tolerant. But those without 1 kilobase per insertion, as you can see here, they are susceptible. So we wanted to check that if there is this kind of insertion in Ethiopia. And we also identified from their paper that there is the majority of the 1 kilobase per insertion is found in the Southeast Asia here, but in Europe, the multi-erythrotransposer methylation gene in the HVS gene is the mechanism for the acid tolerance in the Europe. But as you can see here, there is nothing for Ethiopia. There is no 1 kilobase per insertion. There is no multi-erythrotransposable element in the barley collection from Ethiopia, from few land races which is identified. And we increased the number, and we searched if there is the presence of any of this gene, but we were not able to find. So the HVST1 is like it shows that there is a presence of the HVST1, which is responsible for sequestration or releasing of citrate by the root tip, but it's not functional among the collected land races that we have, all the 200 barley accession, and the other, we didn't find anything. So for that, we proceed breeding using the land race from Japan, that is, which has 1 kilobase per insertion, and we wanted to breed it with the Ethiopian barley. As you can see from this picture, the gel image, this one in the red is the Ethiopian land race. It shows clearly there is no 1 kilobase per insertion. But the Murasaki mochi here, that we use it as a parental material, it has 1 kilobase per insertion. So when we breed the Ethiopian land race without the insertion or the 1 kilobase per insertion and the Murasaki mochi, we get the F1 with both bands are clear present. And then we use the F2 generation and segregating. As you can see, the majority of the F2, they don't have the 1 kilobase per insertion, but some of them, they have 1 kilobase per insertion. So we selected all those barley with 1 kilobase per insertion, and we provide that to the research institute for further validation and selection based on the trait that they really wanted for breeding. So I am happy that we started this with one of my master student, Ayalnesh. Now she is working in the university in the northern part of Ethiopia, where she was able to learn all the molecular tools and become able to publish with this paper. And we are really happy with that. And the other one we did is from our collection of 200 barley, we perform agronomic practice with the agronomy group in our college, and we identified that extreme varieties are there. Some of in the left that you see, the HB was 1307 and 217166. This is the selected barley from the 200 that we have selected from the southern part of Ethiopia. They are found to have higher grain yield, a better straw yield, white color in the case of the first one, but better color, not black, for the second barley. And when we give them to the farmer to select, they were happy to find that with higher grain yield a better straw yield and white color, and specifically adapted to that particular acidic soil environment. They are happy, and now they are popularizing this one. But we don't know what the mechanism of acid tolerance for this. But for example, the other one, 4 and 5, they are poor in grain yield, and they call it poor appearance because the majority of the farmers, they are not happy with the black color of the barley, because it doesn't fetch for them a lot of price when they are selling it to the in the market. So we popularized this one, but we continued to understand what is really makes this barley to grow in that particular region in Ethiopia in acidic soil. And again, with our collaboration, we tried to see the presence of the relative expression of this HVA stage 1, which actually exudate citrate. And we identified, comparing with Murasaki mochi, highly tolerant, constitutively releasing citrate under acidic or non-acidic condition, and Morex, which is susceptible to acidic soil. We have identified that two of the popularized variety, they have significant expression of this HVAST gene 1, which is found in barley chromosome number 4. So there is no insertion, or like it doesn't have a mechanism of 1 kilobase per insertion at the transcription start site, or which happened in Murasaki mochi. It's not there for the Ethiopian land races, but still it is able to transcribe this gene under acidic condition, and also constitutively too. So that means we define that there is a separate or a different mechanism for the Ethiopian barley, which is not similar to the Ethiopian barley, the European barley, or it's not similar to the the Northeast Asian barley. So there might be a unique mechanism of acid tolerant from Ethiopian barley. So now we are working with one PhD student, we are working on breeding and developing a QTL map for this gene. In the near future, we will report to you that. So, and then the last slides, I just wanted to talk about how the traditional breeding affected, because we breed acid-tolerant barley from Murasaki mochi and also Ethiopian line. And Murasaki mochi, with the color, it is a violet color. The seed color is violet color. And it's not, the farmers are not happy to take this violet color seed because they can't sell it into the market. And the market, many people, they prefer white seed. Okay, so as a result, even if we were able to breed it, so we have to rebreed it in order to have the acid tolerant trait, and at the same time, we have to have a white seed for the farmer because that is their preference. And also the hairlessness is not that much accepted by the farmers, and specifically the violet color. So the acid tolerant is really interesting. So this took us a lot of years, like around 8 years, but we are thinking about to jump into the current technology with the genome editing with CRISPR-Cas9, because it focuses only, it takes only the Ethiopian landrace and checking the genome, and then only use the laboratory to develop to edit the gene in the acid tolerant, and by comparing acid tolerant and the susceptible. So we can change that, which only takes a few DNA later. It takes 3 to 4 years, and it is precise, low cost, and predictable. So because the demand for seed by the farmer, they really need it, but we are not able to give them until we are now selecting and breeding. So that is the obstacle of traditional breeding. Even we are happy that we use molecular markers to select that. So I have a chance to get some training from the Flanders Institute of Biotechnology at Ghent University, where I was able to learn about how the CRISPR-Cas9 system works and how long it takes. So within 3 months, I was able to learn that this technology, not on barley, but other plant where the laboratories that have been affiliated were working. So this tells us that the adoption of technology, marker-assisted technology, genome, and also genome editing technology might help us to accelerate the breeding of acid-tolerant barley for Ethiopia and also for the other part of the world. And understanding the mechanism, it can enable us to speed up the breeding process. So why we have to focus on barley to develop this next technology? First, barley is not new to Ethiopia. All knows it from child to adult. From the triticale species, barley is diploid, so it will not be difficult for us to use it as a teaching material or as a model plant for my students. And since there is a well-developed molecular tool, so we can adopt that to improve the barley lines or the barley land races for Ethiopian. This will enable us to teach students from undergraduate to postgraduate level. So that is why I stick on the barley for future research too. So the take-home message from this seminar is there is diversity and it has a lot of potential. And the Ethiopian barley exhibits a remarkable diversity. And some varieties are naturally tolerant to acidic soil. And still we didn't know that. We have going to unravel that. And underlying mechanism of this tolerance remain unknown. We have to unravel it. And the current breeding challenges, specifically with climate change, traditional breeding is not a use. We have to, it's proven inefficient in addressing soil acid tolerance. So advancement in gene editing, efficient gene editing and base editing techniques are available, and in the majority of plants has been done, but we don't have that technology here in Ethiopia, and specifically in the southern part of Ethiopia. We have to introduce that technology for the future. And the use of molecular marker, specifically the whole genome sequencing, that is how it develops the dark sequencing technology. It enables us to understand where the diversity is there, or if there is a similarity among the barley lines, and it is really important to invest on that. So at last, I really wanted to acknowledge a number of institutions who helped me and taught me different lessons. One is from Okaima University, the Institute of Plant and Stress Resources, and the Belgium University, where I was partnering with Thomas Jacobs Laboratory, a wonderful laboratory which enabled me to learn the new technology, and might be in the near future that I will have a lot of partnership. And all the students who are working and trusting me and selecting barley lines, breeding barley lines, and learning from that, and providing that to the farmers. And I am really happy with the farmers, and I'm thankful for the farmers because they gave us their land to cultivate our barley. Thank you.
Thank you so much, Hewan. Very, very interesting presentation. Maybe if you can stop your sharing the screen. And yes, please, everyone who has questions, post them in our chat box. I'm amazed that the uses that you describe for barley in Ethiopia, when here basically it's mono use, I mean beer, that's it. I'm very amazed and interested now in learning more about that. I'm gonna open the questions with something that came across my mind. Is all this diversity already collected and preserved, like in your in the National Institute in Ethiopia or ICARDA? And the reason why I'm asking this is if you succeed, one of the worst situations that I've seen is that breeders are very successful, and because they're very successful, they wiped out the diversity in the field, and that's not a good outcome. So that's why I'm asking about this diversity, whether it is already preserved, whether there are needs to fill gaps in terms of collection and preservation.
Yes, thank you very much for the good question, Carlos. The Ethiopian biodiversity actually is collecting and preserving this diversity, and anyone can access from the biodiversity, but not every time they replace that into their system. And yes, the diversity is kept there. It is
Okay.
well kept here.
Okay. We have a couple of questions from Gina Velasquez.
Mhm.
Uh, what challenges have you encountered when translating molecular findings into field results? That's one question, if you want to answer that first.
Thank you very much. Yes, a lot of challenges we have really encountered, specifically coming from Japan, where I have experienced advanced technology, where I can order a primer today and I can get it tomorrow. But and also the laboratory consumables, even like Notac polymerase, DNTP, or agarose, or everything. That's not a luxury here.
Mm.
So that's why I was succeeding with this research, because I was a support from the Okayama University, specifically from the Institute of Plant Stress and Resources with Professor Sato, and also now with Professor Hisano. They're supporting us, and without them, I was not able to work at all, because you can't get such kind of consumable system, and we don't have such kind of system. So that was the biggest challenge for me.
Mhm. Okay. How, also a question from Gina, how has your research influenced local farming practices or farmers' decision making? And I will put it also in a two-way avenue, how your better knowledge of how farmers and consumers use barley has influenced your research.
Okay. How my research influence the farmers, and also how the farmers' knowledge influences my research. Definitely, it was give and take. Definitely, I never thought that Ethiopia has a lot of diversity, and specifically with the farmers. We went to the southern part of Ethiopia with different language. You have to remember that the region where I belong, they speak Sidama language, and if you go further 25 kilometers, there is Oromofa language. Another 30 kilometers, if you go and you find Alaba language, then it's like in the southern part, there are about 55 language totally, and 55 different mindset, different culture. So all of them, they have their own way of preserving barley, keeping in their own garden, and passing it from one generation to the other, because that was dear for them. And one of the things that I really impressed, specifically when I was working with one of the ethnic group, is that when I give them different type of barley, immediately they identify what that particular barley is. I was not trained to identify that particular test, but they have that
Mm.
experience. So it gives me a lot of information that the farmer are really knowledgeable. So when I take, for example, Murasaki Mochi in their field, and they were like, looking at that, the 6 row, it's very good. The only disappointment for them is it is hairless, because for the Ethiopian farmer, they have to keep their seed in a traditional system of seed storage. So some of the pests will not affect them if it is hued. But in the hairless one is their disappointment. So all this tells me everybody has their own preference in selection.
So I wonder, Hewan, based on what you described, if because one of the questions is what are your next steps, particularly when you consider gene editing. My question, with all this knowledge that farmers have and all this diversity that they are selecting, wouldn't you be well positioned to create diversity using the Japanese line and some of the local materials and give them to the farmers for them to select, segregating populations? It's sort of a participatory breeding approach.
Yeah,
Because it
that is
looks like
actually
in that way you will be introducing improvement in terms of acid soil tolerance, but also giving them the ability to select strains that adapt better to their local conditions and the preferred uses. I don't know. That's something that would be interesting to me.
Yeah, actually, that is what we did, Carlos.
Okay.
What we did is like, after breeding the Ethiopian landrace, it is 6-row white barley with Murasaki mochi, the landrace, which with 1 kilobase per insertion, which is acid tolerant constitutively, which has been proven by different researchers. So we showed them, we planted them in the garden, and they were happy, specifically with the 6-row, and the straw is big. That is also for the farmers, they wanted the seed and also the straw, because the straw is also more valuable. They feed it their animal and also they use it for their house. So they selected that. So that is what I did after selecting, inserting with the breeding. We selected all the lines which has a 1 kilobase per insertion for the HVST gene, and we gave that seed to the research institute. Why I gave it to the research institute? Because like the research institute, they can develop an activity every time they can get the fund, and they have also the land that they can bring the farmers to show it. And after that popularization, they can give it back to the farmers. So I already gave it, and now I think they are popularizing it.
Yeah. Have you another question here? Have you looked into differences between the white nutritional differences, the white and black
No,
strains?
no, we haven't done that check actually. Yeah, I think
Okay.
it's necessary, but that can be done
Mhm.
after fixation of the 1 kilobase per insertion after 5 or 6 generation. So they will select, and what they do is they do the nutritional analysis, and
Okay.
they release the variety based on that information.
All right. A couple more questions before we stop. Your assessment includes preferences in terms of sensory evaluation of new varieties, taste, texture, and aroma.
To be honest, we don't have those technologies here
Okay. So you
because
just give them to the farmers for them to evaluate and see if they
yeah,
like it or not.
yes,
Okay.
yeah,
Okay. Okay. Yeah. So that sort of answered the next question, that your selection criteria is aligned with the farmer selection criteria. Okay. So thank you so much, Hewan. I would reach back to you later on to see if I can have a copy of some of those photos that you showed, showing the
I'll
diversity of uses for barley in Ethiopia. They're very, very interesting to me. Okay. Okay.
definitely I'll share it to you.
So let me share my slide here. Thank you for your participation from wherever you join us. Thank you, Brandon, for your help in organizing the seminar. We will meet again at the same time next Thursday for the 5th seminar of this series by Dr. Saurabh Singh from the Rani Lakshmi-based Central Agricultural University in India, talking about insights into male sterility systems and accelerated vegetable breeding. As always, you have the link to the recording for this presentation in tomorrow's early announcement of the next seminar. You can look at it or share it, okay? Thank you so much again for your participation. I hope everybody has a very good weekend, and we'll see you next Thursday. Goodbye.